Well, good morning, good day, good afternoon, good evening, wherever you are located. It's very early here in the morning at my end in Arizona. I'm Volkmar Weisig. I'm the president of the World Mitochondria Society, and it's my pleasure to co-host this webinar together with Marvin Ideas, who is the founder of the World Mitochondria Society. And this webinar is a collaboration between the World Mitochondria Society and targeting extracellular vesicles for obvious reasons, as if you look at the title, both are coming together. I mean, mitochondria and extracellular vesicles. So Marvin, perhaps you can introduce one of our speakers. Marvin.
Yes. Hello, everyone. Do you hear me? Do you hear me?
Yes.
Yeah, I think I have. Yeah. Hello, everyone. Thank you very much, Volkmar, for the introduction. Good morning, good afternoon, everyone. Really, it is a great pleasure today to have this third webinar organized by the World Mitochondria Society and the Extracellular Vesicles group. It's a great pleasure to have two really outstanding speakers for today. It is so difficult to ask a specific question to answer in 40 minutes or 30 minutes. So the idea today really to answer this intriguing question concerning EVs, exosome, and mitochondria content. First, I would like to introduce, I thanks Carla Lopez, and it's a pleasure to introduce you first for this webinar. We met last year in a meeting in our first meeting in exosomes in Malta. You are coming from University of Coimbra, Portugal, and you are highly specialized in mitochondria biology and EVs. You highlight the interest in the intriguing or the interplay, the communication between mitochondria derived vesicle, EVs, and the cellular health and pathology, biology, if you want. You make a significant contribution in understanding how EVs contribute to mitochondria quality control. It is very important to talk about quality control and EVs and to have this connection. Your work, push the border for, especially in the neurodegenerative disease and cardiovascular disease, immune system, etc., etc. You are a leading expert in mitochondrial dysfunction and vesicle-mediated mitochondrial clearance. Your research offered many insight in the molecular mechanism that underlie various diseases. This is again to highlight that it is more research and translate to clinical application. And we try and you try to answer the critical question is how EVs, how extracellular vesicles mediate the exchange of mitochondrial components, especially in the context where intracellular degradation pathways are impaired. So it is very intriguing to understand this correlation, this communication between mitochondria, EVs, and the host. Thank you very much.
Well, and it's my pleasure now to introduce Pascal. Dr. Pascal de Acunzo is a research scientist at the Nathan Klein Institute of Psychiatric Research in New York. And he is also a research assistant professor at the New York University Grossman School of Medicine. In his early years, he was working in the laboratory of Professor Sassoni at the Tor Vergata University of Rome. During that time, he studied the pro-autophagy protein AMPA1 and its role in mitophagy. He then moved to New York for his postdoctoral training in the laboratory of Professor Levy, who is a world-leading scientist in the field of extracellular vesicles in neurodegenerative disorders, in order to study the role of mitochondrial exocytosis in Alzheimer's disease and Down syndrome. Dr. Acunzo's current research investigates the physiological and pathological functions of mitovesicles, specialized which is a specialized subtype of mitochondria-derived extracellular vesicles, which are found in the brain parenchyma. And before I give the floor to our two speakers, I would like to mention just to all participants, please open the chat box. And in the chat box, you can type in questions, and depending on the time we have at the end of a seminar, we will pick questions which we will ask the speakers for them to answer. And if there are many questions, then the speakers will be able, I hope so, to answer these questions via email. And now, no further ado, and I give the floor to our both speakers. Please, it's all yours.
Okay, so thank you so much, Marvin, for your kind words. It's a great pleasure to be here. I'll share the presentations. So, is it visible?
Yes.
Okay, so as Marvin said, I'm going to try to show a bit how these two, the extracellular vesicles and the mitochondrial come together, and how we can isolate and characterize them. So there are several extracellular particles, vesicle and non-vesicle particles, but the extracellular vesicles term refers to the particles that are released from cells and delimited by a lipid bilayer, and they cannot replicate on their own, meaning that they don't have the functional nucleus. So the size, the mammalian cells size range from 5 nanometers to 5 micrometers, and they release, as you can see here, a very heterogeneous panel of particles, vesicles and non-vesicles. So since they overlap in size, sometimes it's quite hard to differentiate them, to know which are which. So luckily, the recent advances in isolation and analytical methods have led to identification of several of increasing number of these particles, particularly the supermer and the hexamer that were identified more recently, and they were shown that the contact, the mRNA contact and the protein contact, they overlap with the contact of exosomes. So perhaps we are isolating exosomes, but and co-isolate these two particles. So historically, these UVs or these particles was thought to be part of a mechanism, of a cellular mechanism that was related to dispose the unwanted materials to maintain the tissue homeostasis. Nowadays, we know that this is more complicated, and UVs are important players in the intracellular communication, and they can modulate through their cargo the cells, the recipient cells. So in the last few years, this research field has grown a lot. So the International Society for Extracellular Vesicles created a consensus, a manuscript, a paper where it tries to give some definitions to help us characterize these particles. So this minimal information for studies of extracellular vesicles defines several subtypes of EVs. You can see here these small and large EVs that are based on the diameter, meaning that the particles that are isolated under 200 nanometers, they are considered small EVs. If they over 200 nanometers, so the cutoff of 200 nanometers defines this type of EVs. They also can be categorized or defined based on the biogenesis. For instance, exosomes, they are originated from the endosomal system, and they are small EVs, they're smaller than 200 nanometers. But for instance, ectosomes and microvesicles, which are larger EVs, they originate from the plasma membrane. Then we have other specific cellular process, and I can go back. For instance, during cell migration, there's the formation of the microsomes, or during programmed cell death, it can be the formation of these apoptotic bodies. So there's a wide range of subtypes of EVs. How does these mitochondrial components enter into these, go into the extracellular space or enter into these extracellular vesicles? So there are several theories. Some of them are better described than the other. So perhaps the most studied particles or vesicles are the exosomes. So they originated from the inward budding of the endosomal membrane to generate interluminal vesicles that will maturate and form these multivesical bodies and fuse with the plasmatic membrane and release these exosomes. So the MDVs, they are small vesicles that are formed in the mitochondria. They bud from the mitochondria and they are produced in a steady state or induced under stress conditions. And they can fuse with these endosomes, particularly if there's a problem with the transport of these MDVs to the lysosomes for the degradation. So there's an increased fusion with these endosomes that will follow this endocytic pathway and release into extracellular space. They also can fuse with amphisomes and be released into the extracellular space. Then we have larger particles, such as the microvesicles, as you can see here. So these microvesicles, they bud from the plasmatic membrane, and they can carry larger fragments of the mitochondria or the mitochondria per se. Then we have another pathway that has not yet been completely described and unraveled, so it's more difficult to describe. But we know that MDV, the mitovesicles, they have different properties from the canonical EVs, and so suggesting there's a different biogenesis and different biophysiological properties, mechanisms for the generation of these EVs that are different from the exosomes and the microvesicles. And by last, we have these exophore, which are large vesicles. They have up to 4 micrometers, and they are released when the cells are under metabolic stress or dysfunction, autophagy dysfunction, or inflammation, and they can carry this damage. They were described to carry this damaged mitochondria. So all of this process can crosstalk with the EVs and increase the cargo in the mitochondrial components. So getting back to this paper, the guidelines that MISEV established, so they advise to characterize them due to the heterogeneity of the EVs. The MISEV suggests some putative protein markers. So they suggest to assess the proteins that anchor these vesicles to know which are our samples are enriched in which type of vesicles. Also the contact, which kind of proteins we have inside, what is the cargo of these EVs. It's also important to assess for contaminants, meaning that most times we can co-isolate other structures, organolar structures. So it's important, lipoproteins is important to assess the purity of our sample. Also, it's good to know when we're characterizing our sample, which is the possible intracellular origin. And here appears a mitochondria. So they suggest some proteins, some mitochondrial proteins to evaluate the presence or not, to see if they can have some mitochondrial components. And by last, most of the times we co-isolate other proteins that it's also good to characterize and to know which our sample, if it's enriched or not in this co-isolate. So, but it's hard to define based on these protein markers. So the field is growing a lot, but most of the paper rely on some protein markers that define some extracellular vesicles. So as you can see here, the classical exosomes, the small EVs that have under 200 nanometers, they are mostly enriched in tetraspanines, CD63, CD81, CD9. We have the large microvesicles, the oncosomes, that are over 200 nanometers, and they usually are positive for annexin. They have annexin is present in its cargo. And then there's plenty of them. Each one was being characterized as have specific proteins. I'll just refer these autophagic extracellular vesicles were described more recently. And as we can see, they have positive for LC3 and P62. So, but how do these mitochondrial components enter the extracellular vesicles? So we know that mitochondrial dysfunction occurs with age, with different disease conditions. We also know that mitochondria has at least 4 main mechanisms of quality control. So whenever the damage is small, we have this intraorganellar proteostasis that try to maintain the homeostasis of the mitochondria. If there's a misfolded protein, the UPS system is activated. Also, the mitochondria can generate these MDVs. So these, in general, they have been described in response to oxidative stress stimuli, and they are budding from the mitochondria, and they are target for the organelle for degradation. Whenever the stimulus is the damage is too intense, of course, there's no rescue, and the mitochondria is target for degradation. So, but what are these MDVs? So there are different subtypes of MDVs. They are being characterized or defined according to two parameters: one, the membrane, and the cargo. So we can have these MDVs with a single membrane. They are enriched in the outer membrane proteins, and they are positive for MAP2 or TON20, and they can be target for peroxisomes or enter the endocytic pathways and go for degradation in the lysosomes. Then we have other types of MDVs, these double membrane MDVs that have the inner membrane, but also have content from the matrix. And they are being described to be pH positive and also can enter this endocytic pathway or go to lysosome for degradation. So at some point, these MDVs can be exocyted into the extracellular space and being considered mitochondrial-derived EVs. So the biogenesis of MDVs starts with the initiation event that induces a curvature in the membrane, followed by a tubulation and the remodeling of the membrane. And it's finalized with the decision of this MDV that is finally target for the organelle for degradation. So different players have been described in all this process, and they are associated to the different subtypes of MDVs. So importantly, how could these MDVs enter the fuse with the EVs? Once the MDVs are formed, they are target for their final destination. They can be cleared in the cells by several mechanisms. So far 3 have been described. So we have a snare-mediated fusion with the autolysosome pathway for degradation. We have the MDV uptake by the multivesical bodies but it's not well described, this process, or the MDVs can be recruited by the Tolley parking complex and enter the endolysosomal sorting. So all this process, or just some of them, can contribute to these mitochondrial specific EVs that are being released into the extracellular space. So there's no specific method to isolate mito-EVs. What we have is the methodologies to isolate extracellular vesicles that we can use to isolate these mito-EVs and further characterize, as I previously said. So we isolate these extracellular vesicles from the conditioned media, from biofluids, from tissue, and there's different methodologies. We can do a PEG-based precipitation. We can do a differential ultracentrifugation, perhaps is one of the most used. Filter concentration, density gradient by saccharose, size exclusion chromatography that has gained relevance as a method to isolate EVs. We can have columns with precise size, so it makes our sample more homogeneous in terms of size. You can also have immunoprecipitation or affinity precipitation, or last, asymmetric flow fractioning. So all of these methods have advantage and disadvantage. So it depends also on the methods that are available in your facilities, in your institutes. But I want to highlight this graph because it shows if you want a sample that is pure, you should go to choose a specific method. If you want to recover a lot of EVs, you should choose another. So in this case, as you can see, the precipitation, the PEG-based precipitation, there's high recovery of our sample. There's a lot of particles, extracellular particles, not necessarily EVs, because we co-precipitate other particles beside the EVs. The same for filter concentration. On the other side, we have the immunoprecipitation that is based on magnetic beads and antibodies for a specific protein in the EVs. So it's highly specific because we will only bind the vesicles that have this particular protein, but the recovery is very low. In the middle, I think we have a good compromise between the recovery and the specificity. So we have the size exclusion chromatography and the differential ultracentrifugation that gives a good yield, and also the purity is, the compromise is good between these two parameters. So this is a protocol that we use to isolate. We isolate large and smaller from the same sample. So this is from conditioned media. From in vitro cells. So we do first centrifugations to eliminate the debris. Then we do a 20,000 G centrifugation for 30 minutes to obtain these larger vesicles. And then we clean them we do a ultracentrifugation just with PBS to have a cleaner palette. And then we process them for number, size, DNA quantification, protein assessment. The supernatant that we obtain from these large vesicles, we filter just to eliminate with the 200 nanometers filter to eliminate some microvesicles that may exist. And we do 2 consecutive centrifugation to 100 Gs and to obtain the palette. And we proceed as previously. So it's also good to have these, the large and smaller, to compare them and to see what is in each. So it's also, when you characterize your samples, you also have to have the number of particles and the size, especially to know which subtype of EVs you are obtaining. So the measurement of EV size, the diameter, usually is realized in assumptions such as sphericity or mobility. So the methods that are widely used is dynamic light scattering, nano-tracking analysis, resistive pulse sensing, and flow cytometry. From this, I would believe that DLS and nano-tracking are the most commonly used, although they could have a small disadvantage because they rely on the hydrodynamic properties of the EVs, which can give a small overestimation of the size when you compare to the imaging techniques. So, for instance, SCRIO-IM, which is the most accurate method, but unfortunately to measure when you have a lot of samples, it's not feasible to use this technique to measure a huge number of samples. So, as I said, there are different the mitochondria is released to the extracellular space as in different forms, in different conditions. So there's diverse mitochondrial contexts within the EVs. We have mitochondrial proteins, we have mitochondrial DNA, RNA that may exist in these EVs, and we can have different methods to quantify them: Western blot, quantitative PCR. We can also have probes, TMRM, MitoTracker. We can also have a multiomics approach to assess the to do lipidomics, proteomics. Also, I think it's important to do functional assays to evaluate the impact of these mito-EVs on the recipient cells. We can do dynamics, we can study bioenergetic and see how these mito-EVs are impacting the recipient cells. So I'm going to show a few studies that these use the same protocol as I described. So they isolated large and small EVs. They were characterized for the protein contact and also analyzed for some inner membrane mitochondrial proteins and outer membrane. They didn't find any in the small EVs, but they saw an enrichment in these large EVs. Also, when you inhibit the acidification of the lysosomes by the use of buffelomycin, you see a huge increase in the number of EVs that are released, but also in the mitochondrial content as expected. So when they performed the proteomic analysis, they found that there was a large number of proteins that were present only in these large EVs, and they were associated with membrane-bounded organelles, including mitochondria, but it didn't find that in the small EVs. So this is some studies that we perform also. So we induced, in this case, we characterize for the proteins markers as normal. And then in these cells, we induce mitochondrial and lysosomal dysfunction to have CCP and chloroquine. And we saw a huge increase in the number of EVs that they are released. We next look in cells that we already had characterized, in this case, PD cells, that had the mitochondrial lysosomal dysfunction. And we also found a huge increase in the number of EVs that are released. And very interestingly, although the number of the mitochondrial DNA copy number was decreased in these cells, we saw an increase in the copy numbers that are released in these EVs. So another method, I'm not going much into it because Pascal is going to talk more about this, but I think it's also an interesting method to fraction to give us this subtype of EV. So you can use, you can separate particles based on the density or size using the centrifugation. So we'll get all these fractions that you can further characterize, in this case using iodixanol to perform this separation. So another method that I think is used to evaluate this crosstalk between extracellular vesicles and mitochondria is the imaging techniques. So as you can see here, these cells were expressing CD63 vesicles, and they were co-cultured with the cells that the mitochondria were labeled for DS red. And you can see the migration of the vesicles toward the center of the other cell, the soma. And here you can see the colocalization between these CD63 positive vesicles and the mitochondria. So I think it's interesting also to analyze this kind of crosstalk. So previously I've shown a study that only the large vesicles had mitochondrial proteins. And this study is from our lab, and we characterized different cells, neurostem cells, fibroblasts, and EVs that we collected. We perform the normal characterization. You can see here the the electronic microscopy, the bilipid layer of the EVs. And what we found is that these small EVs also carry mitochondrial proteins. We found several mitochondrial proteins within these small EVs. So it seems not only the large, but the small EVs can carry these mitochondria proteins. So we also were interesting because we found an increased amount of mitochondrial DNA within the EVs. So we ask ourselves, is it intact? Is it degraded? Is it, so we look for several fragments within these EVs. And from the, as you can see here, the 16,000 base pairs of the mitochondrial DNA, we found small and large fragments. So it seems that the mitochondrial DNA that it's internalized in these EVs, it's somehow preserved. It's not completely degraded. So last, but not less important, I think it's also important to evaluate the function. So in this study, what the authors did was evaluate if these mito vesicles have intact mitochondria. So they labeled the cells with the EV, with the mitotracker and calcine, and they saw that almost 70% of these mitovesicles, mito-EVs, they were positive for these dyes, showing that they harbor intact mitochondria. Also, the larger vesicles, they were positive for the mitotracker, while the smaller ones were negative. So the larger vesicles will carry these intact mitochondria. Another important aspect is if those mitochondria are functional or not, because it will impact the recipient cell's function also. So they perform TMRM labeling of these cells to assess the polarity. And what they said, although it's very dispersed, the results that a medium of 30% of these mito-EVs, they carry mitochondria with polarized and functional mito-EVs. So in conclusion, I think the mito-EVs are a very heterogeneous population with distinct mitocargo and with a wide range of functionality and biological distinct roles that need further clarification. So I'll pass it to Pascal.
Okay.
Share?
Can you see my slides?
No.
Do you see my slides?
No.
No.
No? Oh, why?
You can wait
Okay.
1 minute. Oh, yeah.
Let's see. Better?
Yes, now you can see.
Yes.
Okay. Okay, thank you very much, Carla. It was very interesting presentation and data on mitochondria-derived EVs in vitro. So on essentially on cell culture media and these kind of sources. In our lab, we've been, so we'll switch a little bit gears. So in our lab, what we have been studying was in general extracellular vesicles in the brain. And we do isolate EVs both in the brain of from the brain of mice and human tissue, and of course, you know, the analysis of mitochondria-derived EVs in vivo, so isolated directly from the tissue. So I will talk today a little bit about why EVs are important, why we should really pay more attention, I think, to EVs in solid tissues. I will give you essentially an overview of the data we obtained by our new method that we developed in our lab, in which we are able to specifically isolate mitochondria-derived EVs without contamination of other types of small EVs, at least this works for small EVs. And I will describe a little bit some of the characterization we have done both in normal, physiological conditions and in some condition of disease. And of course, an example of, how to use this characterization and how this type of analysis can be useful in the study of neurodegenerative diseases. And specifically, I will talk today about Alzheimer's disease and Down syndrome, but we published several papers also on other conditions of mitochondrial dysfunction, for instance, the condition of chronic exposure to cocaine, which is known to induce mitochondrial dysfunction in the brain. So I don't have to go in details on all the subtypes of EVs that exist, because Carla very nicely gave us a good and thorough overview. What I'm going to stress here is why it's important to isolate EVs from the brain, from the tissue, okay? So we know that EVs are very important for cell-to-cell communication, are important in mechanisms also for waste disposal. So whenever you have lysosomal impairment, as Carla showed, for instance, when you give chloroquine, you see this huge increase in EV release. Not only mitochondria-derived, but also other types of EVs are strongly, their secretion is strongly stimulated. And so are important also for, as an evolutionary mechanism, essentially to survive, right? So to eliminate material that otherwise would accumulate in the cell if the lysosome is not working. And of course, by shuttling from one cell to the other, so sharing material, they are able to regulate responses in recipient cell, and via cargo, for instance, microRNA, they can modulate the gene expression in recipient cells, as well as immunity and inflammation. I don't have, again, to stress to this audience the importance of the regulation of bioenergetics also via mitochondria-derived EVs, as been described also by other groups. But why, and let's go back to the topic, why they are so important? It's so important to isolate EVs from the brain. Well, first of all, there are several reasons. One of these reasons can be for diagnostics. So by comparing, and by this characterization that we are describing, so by comparing a disease state versus a physiological state, it's possible to find new biomarkers that can be used, can be valuable biomarkers, which have not been still in clinics yet, that can be used in CSF, cerebrospinal fluid, and plasma of patients to diagnose diseases. They can be important as therapeutic agents. As we described, mito-EVs, particularly, they can regulate bioenergetics, and we know that bioenergetics, and metabolism in general is altered in aging and neurodegenerative diseases, virtually all of them. And another thing is that isolating EVs from the tissue gives you a platform for further bioengineering. This is because EVs, they contain essentially proteins on their surface that can drive the tropism of these EVs specifically to the brain. So this is an advantage of tissue EVs as compared to cell culture. And of course, another technical aspect is that the yield of EVs isolated from the brain, given that you have billions of cells in a small volume, essentially, as compared to cell culture, in which cells grow flat, and so you need even in a larger area, you don't have so many cells as compared to the brain, in which you have a multidimensional structure. So the yield is much, much more higher in the brain as compared per milligram as compared to cell lines. So, given this, our lab has been pioneering the study of and the isolation of extracellular vesicle from the brain. I'm not going to go in detail in this protocol. We published it with a very thorough protocol. If people are more interested, you can find it on Nature Protocols. We published it 3 years ago. But a very quick overview. Essentially, what we do, we digest the tissue in an enzyme which is called papain. Papain is known to be a very broad spectrum protease that essentially what it does, it's cleaving the extracellular matrix, so releasing the fluids and releasing the EVs that can be binding to the extracellular matrix. And after that, we do a series, as Carla described before, a series of filtration and centrifugation steps in order to eliminate cells, myelin, and all other kind of possible debris that we can have. At the end, what we do is performing a sucrose gradient to further purify contaminating proteins from vesicular components. And so, as you can see, what we get, it's essentially after the gradient that a pure population of extracellular vesicles as shown by transmission electron microscopy. When we analyzed these EVs by Western blotting, but also by mass spectrometry, actually, the first evidence was via mass spectrometry, we did identify a lot of mitochondrial proteins. And so this is in line with other in vitro data in which they show that mitochondrial material is actually exocytosed and released via EVs. This is just an example. As you can see, we can identify, we can identify the protein PDH1 alpha, which is actually an important protein, as Carla was mentioning, is one of the markers that is used for MDVs. In the fractions containing, in this case, for instance, ALIX, CD63, and annexin. So I don't have, again, to stress the possible hypothesis, possible pathways by which mitochondria-derived EVs are released. So we were wondering which one of these pathways is more prominent in the small EVs isolated from the brain. If maybe you have the possibility of pieces of mitochondria wrapped by ectosomes, you may have mitochondrial cargo into the exosomes, or you may have that MDVs themselves or a specific subtype of MDVs is released upon inclusion in the multivesicular body into the extracellular space. So essentially, mitochondrial material in ectosomes, mitochondrial material in exosomes, or a type which is non-exosome derived, non-ectosome derived, but which derive directly from mitochondria. So we wonder which one of these subtypes is more prominent in the brain. And to answer to this question, what we did was to essentially change the final part of the protocol. So we do isolate a crude EV pellet, but after that, instead of doing a sucrose gradient, which is essentially a low-resolution gradient, what we did was to perform a iodixanol high-resolution gradient, so that we are able to separate EVs very finely separating them about very, very small changes in their density. Carla was mentioning before that in the field, the people have been used size-based methods, for instance, size exclusion chromatography, as well as ultracentrifugation is also a size-based methods routinely to separate EVs according to size. The problem with this approach is that, and again, as Carla mentioned, is that several subtypes of EVs, they share the same size. So they are found in the same samples, although you separate them by size. That's why we use a different approach. So instead of separating them by size, we separated them by density. And using this approach, as you can see, we are able to separate EVs that are enriched in the marker annexin A2, which is a classical marker of ectosomes. But in the paper, we showed also annexin A1, annexin 5, all markers that have been published before to be markers of ectosomes. And we can see them essentially enriching in the light part of the gradient, okay? So on the top of the gradient, in this case, specific case, fraction 1 to 3. As you can see, they are enriched in markers for ectosomes. We do show plasma membrane proteins. For instance, we show that the sodium-potassium channel is there, that caveolin is there. So these EVs are really enriched in plasma membrane components, and we do see them in the light part of the gradient. When we analyze the classical marker of exosomes, example CD63, ALIX, in the paper, again, you see much more here. For the sake of simplicity, I only put ALIX as an example, but as you can see, this marker, they peak essentially in the middle part of the gradient, so with a peak in fraction 5, where we have only very low levels of annexin A2, so there is a shift and an enrichment of exosome in the fraction 4 to 6. But very interestingly, when we checked for mitochondrial protein, we did not find that the bulk of the mitochondrial material is found in ectosomal fraction or exosomal fraction. What we saw is that the bulk of mitochondrial material, in the brain, it's found in the densest part of the gradient. As you can see, we do not see here peaking in fraction 8. As you can see in fraction 8, we do not see the presence of residual exosomes or ectosomes. And so These EVs cannot be classified according to that. Also, we analyzed several RAB proteins, which are known to be found in the endosomes and are also marker of exosomes. I was mentioning before about caveolin and sodium potassium pumps. We do not see them in fractionate. So again, we cannot classify these EVs either by ectosome or exosomes because they do not essentially derive from the plasma membrane or from the endocytic system. We do see markers from all the mitochondrial compartments. As you can see, we could find markers for matrix, which is PDH1 alpha, from the inner membrane like COX4 or from the outer membrane like VDAC. And this suggests that you have a double membrane structure, which is consistent with the double membrane MDVs that Carla was mentioning. And we do not see, and this is, I think, very interesting, we use a Golgi marker 130 as a marker to show that there is no contamination of intracellular material. But we also see a lot of mitochondrial proteins that are actually lacking in our mitovesicles. So this suggests that the proteome of the mitovesicles, it's very specific, and it's not just recapitulating. They are not just mini mitochondria. They do not recapitulate the whole repertoire of proteins that you can find in mitochondria, but only a specific subset. For instance, in this case, we analyzed the proteins mitofusin 2 and TOM20, and we found, but again, we demonstrated that many proteins, we analyzed polymerase gamma, we analyzed other type of proteins, ribosomal, mitochondrial ribosomal proteins. They are not found in mitovesicles, okay? So, driven by this, we wanted to analyze more and to characterize more these mitovesicles in the brain. So we did some analysis, some of the analysis that Carla described before. We did the nanotrack analysis or NTA. We did the protein content, and the take-home message of this slide is that essentially the number of, as you can see in the first graph, the number of mitovesicles is extremely low as compared to other EVs, represents around 1% of the total small, we're talking about small EVs, okay? So 1% of the total small EV population in the brain. But they have relatively high amount of proteins. So essentially, whenever we crossed the number of EVs as measured by nanotrack analysis and the amount of proteins, as you can see, these EVs, they contain per single EV more protein than exosomes or small ectosomes. And this is consistent with their density. It has been shown before that the relative ratio between protein and lipids is what drives essentially the changes in the density of the vesicles in general. I mean, in any type of particles, even when people analyze lipoparticles, you can differentiate them between LDL and HDL, low density and high density. And this difference is driven essentially by the amount of proteins per particle. The size. So again, as you can see in green, you can see the ectosomal fractions, which they peak in a later size as compared to the blue fractions, which are the one that are reached in exosomes. So again, we demonstrate even within the small EV population, ectosomes tend to be bigger than exosomes. While when you see the red one, it's essentially in the middle between the two. So mitovesicles tend to have more or less slightly the same size between exosomes and small ectosomes on average. That's why they co-pellet together. We did, and this is also Carla was mentioning, we did the cryo-EM, and cryo-EM is the gold standard in the field, both to analyze their size more precisely, as well as to visualize their native electron density. So the pictures you see here, it's essentially these EVs are not stained, they are not fixed, so they maintain their small round shape. And yes, so they are not stained. So the signal you see, it's inherent to each EVs. It's showing their native electron density. As you can see, fraction 1, 2, 3 are enriched in these larger EVs again, and this is consistent with our NTA data and with their nature as ectosomes. We do see that fraction 4 to 6 contain these EVs with a granular core, very granular. And interestingly, for this audience, is the fact that in fractionate, we do see a very unique subtypes of EVs. As you can see, they have this very, very dark core. And in EM, the color, the dark color, is proportional to the amount of electrons you have. That's why it's electron microscopy, so you see electrons. The more electrons you have, it means that you have molecules with a higher molecular weight. So this suggests that this material may contain, this mitovesicle may contain, similar to mitochondria, they may contain high molecular weight atoms, like, for instance, iron or copper, you know, the one that are known to be bound to the electron transport chain. And we confirm the presence of a double membrane. So we do see one extracellular membrane, an intermembrane space, and an inner membrane. We did the analysis of the proteome, as I was mentioning earlier. The proteome of mitovesicles is very, very selective. Only 24% of the mitochondrial proteome is found there. And we do not see whatsoever any protein involved in mitochondrial DNA replication, transcription, mitoribosome subunits, some assembly factor, or iron sulfur cluster biogenesis and assembly proteins. However, we do see proteins involved in energy production and catabolic pathways, as well as amino acid and neurotransmitter metabolism. And this was validated by Western blotting. As you can see, we did several of these markers we analyzed, and some of them are strongly enriched in mitovesicles as compared to the brain homogenate, which in our case is a surrogate for the intracellular mitochondria in the brain homogenate whenever we analyze mitochondrial proteins. So some of them are strongly enriched, some of them are present, but at very much lower level than when compared to the brain homogenate, and some of them are absolutely absent. So TOM20 I was mentioning to you before, mitofusin, the full length of pink1 is not found. We found this cleaved form, which is pink1 delta 2. We do not find the RP1, and so on. So again, this suggests a very specific proteome in these mitovesicles in the brain. We did also lipidomic analysis. Again, for the sake of simplicity, I'm going to just summarize the results. So what we see is that mitovesicles, they contain a unique lipid content as compared to other EVs, which is reminiscent of mitochondria. So for instance, we do see higher levels of cardiolipin. And cardiolipin is a protein, it's a lipid which is found, as probably the audience knows, it's found only in the inner mitochondrial membrane. So this suggests that not only your material, but the membrane itself comes from mitochondria. Otherwise, you would not find cardiolipin there. We do see also other characteristics that are very typical of mitochondria: lower level of cholesterol, relatively higher level of phosphatidylcholine as compared to other EVs, and the high level of phosphatidylethanolamine, which is also found at higher levels in the mitochondria. So we analyzed if, given that they have a double membrane, given that we saw by mass spectrometry the presence of electron transport chain, complexes, Krebs cycle proteins, and all the machinery required for producing energy, we wondered, first of all, if these EVs could, by themselves, so in vitro, like isolated mitovesicles without a recipient cell, if they could produce an electrical gradient, and if so, if they could produce ATP. So what we did was essentially to incubate them with precursors of the energy ATP production via electron transport chain, essentially pyruvate to feed the machinery, and malate as a precursor as a Krebs cycle precursors, and ADP and phosphate, of course. And then we put them at 37 degrees in presence or absence of different drugs, for instance, FCCP to depolarize them, or oligomycin antimycin to block the electron transport chain 3 and 5. And as you can see, when we stain them with hematotracker DPRED, which is known to stain mostly actively polarized the mitochondria, in this case it's EVs, so mitovesicles, as you can see, we saw a decrease in when we do using this potentiometric dye, we do see a decrease in the number of positive mitovesicles when we give FCCP, suggesting that there was an electron, like an H plus delta psi gradient. And when we did an ATP production assay, we do see that very quickly, in 10 minutes, they can double the amount of ATP present in the sample. but this is not achieved essentially with if you block complex 3 and complex 5, suggesting that ATP is produced. More importantly, we do see the presence of monoamine oxidase A and B, and these monoamine oxidases are very important in the degradation of neurotransmitters like dopamine and serotonin. So we wondered if their activity was preserved in a cell-free system in mitovesicles, and the answer is yes. So we did a monoamine oxidase using several inhibitors. Pargilin is an inhibitor of monoamine oxidase B, and chlorgilin of MAO-A, and we do see that when you give both, you completely abolish the monoamine activity, while when you give single ones, we do see a preponderance of MAO-B activity over MAO-A. So in the last part of this talk, I was just to describe you how we can use this isolation of mitovesicles to infer something about diseases. This probably is not that I think that the audience knows this very well, but essentially Alzheimer's disease, as well as Down syndrome or physiological aging, they're all characterized by the mitochondrial dysfunction. So as well as alteration of brain metabolism. So we thought to analyze mitovesicles in this context to see if this dysfunction in mitochondria could reverberate to the mitovesicles and or vice versa. These are just data on the right side on using our mouse model of Down syndrome, which will be familiar in a later slide, which is called the TS2. It's a model of essentially it's a segmental triplication of some of the genes found on chromosome 21 in humans. And as you can see, similar to this mouse very nicely replicates the mitochondrial dysfunction is found in humans. So we compared the hemibrain here. So it's just a brain homogenate in these mice as compared to human cortex in people with Down syndrome as compared to diploid controls, age-matched diploid controls. And as you can see, we do see an upregulation of the mitochondrial fission factor, MFF, upregulation of the activation of DRP1 via phosphorylation and serine 616, suggesting mitochondrial fission and fragmentation in these mice, similar to what we see in the human cortex. So we did analyze mitovesicles, and as you can see, we do see an increase in mitovesicle secretion. So the levels of COX-4 in this case, whenever you load your EVs by macroliter, you see an increase of it. We did a measurement of the number of mitovesicles via MTA, and we do see an increase in number of mitovesicles in both sexes, both male and female mice. TS2 mice have more mitovesicles in their brain as compared to controls. And this is true also when we analyzed other mouse model of beta amyloidosis. We did this to prove that this is not specific only of Down syndrome, but it can also be found in mice that overexpress APP mutated forms. Or we also did actually in the APP knock-in, which does not overexpress, but still is a mouse model of beta amyloidosis. And in all cases, as you can see, we do see an increase in the mitovesicle secretion. So long story short, both in AD and DS, what happens is that, or at least that's our model, that's what we think it's happening, is that whenever you have mitochondrial stress, and this is paired with an impairment in lysosomal activity. These have been published before by first by the Nixon group, but then validated by other groups also, that in Down syndrome and Alzheimer's disease, very, very early, you do see endosomal and lysosomal dysfunction. So the combination of the two, mitochondrial impairment and lysosomal dysfunction, drives essentially a higher secretion of mitovesicles outside of the brain. Why this is important? Well, because mitovesicles can have activity, they have a gain of function activity once it's in the extracellular space. So what we found essentially is that mitovesicles are able to impair long-term potentiation, only the TS2 mitovesicles and not the control mitovesicles. And all the other EVs are not able to do that. So this is a specific characteristic of mitovesicles. And this can be found also when mitovesicle from, not only from the Down model, but also from the Alzheimer's disease model, the beta amyloidosis model, sorry, were used. So, long story short, in the conclusions, we do have a method that let us separate reach at least mitovesicles, exosomes, and ectosomes from the brain. We have relatively high purity. We do not see contamination of cellular debris. We do not see intracellular mitochondria, as I showed before. We don't see TOM20. We don't see 80% of mitochondrial proteome. So this let us differentiate between intracellular mitochondria and mitovesicles. We don't see other EVs influxionate. And as I showed, we can count them via NTA, and this let us understand the secretion, how the secretion or the uptake, depending on the type of experiment, is altered in physiology and disease. And I also showed that in the last slide, we could use this mitovesicle for functional studies, so to study long-term potentiation, for instance, in hippocampus slices. Of course, nothing is perfect. So also in our case, we have other limitations. One is that we need an enzymatic digestion. So theoretically, at least, papain may alter the composition of the proteins on the outer membrane, although we do not have really evidence for that, but theoretically can happen. The process of producing a column is time consuming. It's a low throughput process. You cannot process so many samples at the same time, that's for sure. And as I mentioned earlier, the yield is relatively low because fractionate mitovesicles approximately are 1% of the total EV population in the brain. I want to thank my mentor, for sure, Efrat, that introduced me to the world of EVs and was very supportive from the very beginning in these studies. So thank you very much, Efrat, and all the other people in the lab, both senior scientists, postdocs, and the technicians that helped us producing the data that I showed you before, as well as our collaborators and our funding agencies, and of course, everyone for the attention.
Thank you very much. Thank you very much, Pasquale. Thank you. Thank you, everyone. Thank you, Carla, also again. So in order to have very selected questions, maybe, Carla, can you selected one of the blog question? We cannot answer all of them. I think maybe you can selected one. And the same for Pasquale, if you can selected one question, and then we have maybe, if you have time, we can selected one more.
Well, one of the questions was about, it's okay if I start, or Carla, you want to start first?
Yeah, of course, I'm still reading, sorry.
Okay. No, it's just, well, because one of the questions I just read here, it's about molecular mechanism that trigger the formation release on mitoEVs. Well, first and foremost, and this has been published by many groups, not just, it's our data, but Carla showed the same, and other groups also show the same. So it's very consistent and very robust. Lysosomal dysfunction seems to, or at least giving a lysosomal block with bifidobacterium, with chloroquine, or in our model, you have lysosomal dysfunction in the brain. It seems to be a strong triggering of the effect. Production of reactive oxygen species. This also has been shown by the McBride group, many papers, and also in our hands. Every time you specifically trigger, for instance, we did an experiment, we just gave antimycin A to the cells, so you block complex 3, you have an increase because there is more proton leak, so you have an increase specifically in the mitochondria of oxygen, of ROS production, and you see a huge increase in mitovesicle release. So, and this again, it's consistent with the literature. So I would say that the two main triggers are mitochondrial alterations, so specifically ROS production, and the lysosomal dysfunction. I don't know if you agree, Carla, but yeah, papers are all concordant in this. Other molecules, DRP1 probably is important, and this is also shown by the McBride group that whenever you block, there were a little bit of controversies initially, but now it seems established that when you block DRP1, you block essentially the vesiculation of mitochondria, and so this probably DRP1, similarly to how regulates mitochondrial fission, also regulates the vesiculation by pinching the protrusion that I created. And am I missing something, Carla? I don't know.
No, yeah, I agree with you. Also, Miro has been involved in this.
Yes. Miro, yes.
MVVs, yes. So,
Carla, if you can select a very short question and answer, please, so we can have a chance to answer other questions here.
There's one that these free mitochondria of these MDVs, these mitovesicles in blood or other body fluids serve as a reliable source of mitochondrial gene therapy, or even if they can be used for other therapies. So I can speak about my experience, because what I found inside these mitovesicles, for instance, in the mitochondrial DNA, is that it's highly oxidized. So most of the complement of, because I also work with the neurodegenerative disorders. So I think this mechanism, as Pascal was saying, that the mitochondrial dysfunction, the lysosomal dysfunction, the increase of ROS, so the components inside the mitovesicles are not in perfect conditions. Perhaps if you go to acute conditions, for ischemia, for instance, when those mitovesicles are from healthy cells, and they can rescue the penumbra where the lesion is. And in that case, I think those mitovesicles can have a benefit on the tissues around. But in our case, in the neurodegenerative disorders, I don't think the components within the mitovesicles are that healthy too. They can use these as biomarkers, but for rescue
about mitovesicle and free mitochondria. So maybe you can answer, or Vortmar, you want to comment about your question?
Who?
We don't hear you.
Happens all the time. Sorry. How can you strictly differentiate between mitovesicles, which have escaped from the cell, and mitochondria, which have escaped from the cell? I understand that mitovesicles don't have all the functions, but I mean, we can have, and this one table he showed, I mean, it's just more or less. So that means, is there a gray zone between when mitovesicles actually can become a free mitochondrion?
Very, very interesting question. So first of all, they don't have crystal. So they are morphology-wise, they are different from a regular mitochondrion. Size-wise, they are much smaller. So I didn't stress this enough, but my mitovesicle is in the order of 100 nanometers. So it's very small as compared to a regular mitochondrion. Third, the proteome. The proteome is very different. I showed here the functions that are retained, right? Like, for instance, energy production. But we all know that mitochondria are much more than that. Mitochondria can do a lot of other things. For instance, mitochondrial, they can translate their DNA. They can transcribe their DNA. They can produce their own protein. And to do that, you need mitochondrial ribosomes, you need all the machinery, you need TFM, you need polymerase gamma, you need all these proteins are not there. So they can be used as a discriminant. And I'm saying, like, the presence or absence of this protein can be used as a discriminant between the larger EVs or larger mitochondria as compared to mitovesicle. Example, TOM20. As Carla showed before, right? Also, the Liang group showed that in large EVs, you do see TOM20, right? So TOM20 is present in a larger, potentially nude mitochondria or ectosomes containing mitochondria, but it's not present in the small one, okay? So that's one of the molecules that can be used to discriminate between the two. But as well as I said, mitofusin 2, for instance, is not present. So it really seems that there is only a very selective pool of proteins that are specifically packed into these small VVs via mitovesicles. And it's likely that they are, as Carla was mentioning, oxidized. So probably the proteins that are mostly oxidized, electron transport chain or other proteins that are more in direct contact where proton leaks happens, are the ones that are preferentially secreted in the outside. Via small EVs. Large EVs is a different story.
It's a different story. Yeah, I agree. I completely agree. What is really interesting, I think, also is that following your question, is that those components of these EVs, they can be endocyted by the other cells, and maybe they can integrate the mitochondria in the other cells. And if they have super complex, if they have some proteins that can rescue or can be integrated in the other mitochondria, I think that can be a mechanism of the cells to preserve itself.
Okay, thank you.
Thank you. I think quickly, if you can answer the question about the immunogenicity of, we have one question about that. I am looking for it. If you can answer it quickly, please. How does EVs, exosome, mito-EVs escape from the immune surveillance? Do they express "don't eat me" signal?
From my experience, and I maybe I can just give a small clarification. So I work mostly with DAMPs, so the damage-associated molecular patterns associated to mitochondria that are within the EVs, and those are highly immunogenic. So whenever I expose microglia to these EVs, I see inflammatory response. From the EVs, if I tried with the mitochondria, the intact organelle, so isolated the mitochondria and exposed the microglia, and there is no response. So it seems that has to be these components that are exposed and that trigger this immunogenic response, at least from the microglia.
Thank you very much. Other question, Volkmar, or I have only the last one? So first, really thanks for everyone. I have very short question for you, and I really, if you can answer me very shortly. You are young, you have energy, you have very nice perspective. So if you can imagine yourself in 5 years, how you imagine the field of EVs in 5 years?
Hard question in the sense that, yeah,
Short, clear, and I need really clear from you. How you imagine? What is your vision?
So EVs field in general has been growing exponentially in the last years. So if you see the number of papers published every year in the last probably 10 years, that's been exploding. So it has to reach a plateau level. It's impossible to keep growing at this rate. So I'm talking about EVs in general. Then if we talk about mitochondria-derived EVs, I think there is a lot to explore, a lot to do. We are still we are still in our infancy in this field. There is so much to study about, and both in any condition. Let's talk about mitochondria EVs in cancer, for instance. There is not much, or in lupus, right? You have lupus, you have a lot of anti-mitochondrial antibody. Nobody knows why. So there is a lot to do in this field, and I don't know if it will happen, but I truly hope that the field will grow as much as other EV fields have grown.
Carla, thank you very much.
Yeah. I completely agree with Pascal. I think the major player here, what will be decisive to advance this field, is the technology, the methods. So first we need consensus, because sometimes one paper or the other, they have different results just because they don't use the same techniques, or they don't. Sometimes it's hard to get conclusions. But I think the technological advances will be the resolution, the capacity of seeing these very small particles and differentiate them from the rest. Being able to really see this subtype of EVs will be essential for the advance. I think it's the next step to move this field forward.
Thank you very much. Just to remind you, we have a meeting on mitochondria next October, and we have also a meeting about EVs and mitochondria macrobiota in October also. So, Volkmar, maybe you can say the final word.
Well, if you want to say me the final word, it's just amazing. It's just amazing, the field of extracellular vesicles. I mean, I heard the first time about extracellular vesicles maybe 4 or 5 years ago, and I severely doubted everything in this field, to be honest. And it's just amazing, this field exploding, yeah. And you gave a very good talk, and I would like to thank you very much. And well, I don't have a question now, but hopefully, eventually, maybe extracellular vesicles will enter the clinic. And then it becomes really exciting. But I mean, it might take more than just 5 years, maybe, I think. But that's another big discussion, which I don't want to start here. So thank you very much.
Thank you very much, everyone. Thank you very much, Carla. Again, Pasquale, thank you very much. And wish you a nice day and nice night. Thank you very much, everyone.